Hi, everybody. So we're going to continue on with our discussion of genetics today. And on the first genetics PowerPoint, we talked about how autosomal traits are inherited. So those are genes that are on autosomal chromosomes, which are chromosomes 1 to 22, and for which everybody has a homologous pair. We talked about a couple disorders that were due to mutations in autosomal dominant genes, most notably Huntington's disease. Today, we're going to talk about a few autosomal recessive disorders that are due to a recessive gene on one of the autosomal chromosomes. This is certainly the more common type of inherited genetic disease. And the reason for that is because if you are a carrier, that is, if you have one normal gene, and in this case, it's the recessive gene that is affected, then because you have one dominant gene, that means you do not have the disease. However, because you are a carrier, that means you have a 50% chance of passing the affected gene onto your offspring. In order for offspring to have the disease, both of the parents would have to be carriers and get the affected gene from both parents in order to have the disease. So with dominant disorders, you can't be a carrier. You either have it or you don't have it. But with recessive disorders, you cannot have the disease, but have one of the mutated genes and can possibly pass that on to your offspring without you having it yourself. So with the for the autosomal recessive disorders, the genotype of someone can be a heterozygote is a carrier. Then a person who has two dominant genes is normal. A person who has one dominant and one recessive is a carrier. And then a person who has two recessive genes has the disorder. So a carrier has one dominant, one recessive. Someone who has the disorder has two recessive, and then a completely normal would have two dominant. So we're going to talk, there are many examples of autosomal recessive disease, but I'm just going to give you two of them as examples. The first one I'm going to talk about is PKU, also known as phenylketonuria, and that's a mutation on a gene on the chromosome number 12. And the other one I'm going to talk about is cystic fibrosis, which is also autosomal recessive, and it's a mutation on a gene on chromosome 7. So we're going to talk about both of those in a little bit more detail to give you an example of autosomal recessive diseases and how they're inherited. This Punnett square illustrates how an autosomal recessive disorder is inherited. In this case, it's for PKU. I'm going to switch to a different color, that green. For PKU. And so this one's not the greatest because the letters look so much alike, but we have both parents are carriers. So they are heterozygote. They have one normal gene and one affected gene. So when they form gametes, the gametes are either going to have the normal or the affected one for both parents. So if the offspring gets both normal genes, they are homozygous dominant. They do not have the disorder. If they get one dominant and one recessive allele, then there are heterozygotes. They don't have the disorder, but they're a carrier. And if they get two recessive alleles, then they're homozygous recessive and they have PKU. And that's pretty much the pattern for all autosomal recessive disorders, that you have to have two recessive genes in order to have the disease. Phenylketonuria, PKU, is caused by, it's a defect in an enzyme. And it's an enzyme that breaks down the amino acid phenylalanine. If you remember from earlier in the semester, we talked about amino acids. They're the building blocks of proteins. There are 20 different amino acids. One of those amino acids is phenylalanine. And phenylalanine we get from food. So when you eat foods that contain proteins, you're getting phenylalanine. Also, if you're breaking down body proteins, but that's not normally the way you would get your phenylalanine. You can break down the protein that you eat. You get amino acids. One of those amino acids is phenylalanine. Phenylalanine is what's called an essential amino acid, which means we have to eat it. We can't make it ourselves. So you have to eat phenylalanine, and you use it to make your body proteins. If you have more phenylalanine than what your body needs at any particular time, the extra phenylalanine you break down and eliminate. In PKU, what the problem is that when you get the phenylalanine from food, you don't have the enzyme that enables you to break down any extra. So that enzyme is missing. It's called phenylalanine hydroxylase. And as a result of that enzyme being missing, any extra phenylalanine that you have, you can't get rid of it, break it down normally to get rid of it, and it builds up in your blood. Eventually you start excreting a byproduct in the urine, and that's where the phenylketonuria, PKU, comes from. It's the byproduct of the phenylalanine that's excreted in the urine. And this buildup of phenylalanine can cause health problems for a child in particular. In particular, can cause severe brain damage. So it can be detected at birth. Newborn babies in this country are all given a little heel prick when they're born, and it's one of a number of genetic diseases you can treat. And so they screen for it at birth. The way that it's treated is by giving a diet that's low in phenylalanine. And the problem is you can't totally eliminate phenylalanine because you need some to make your own body proteins because it's an essential amino acid. But if you have too much is when it builds up and causes problems. So they have to have a very regulated diet. There is a formula that's available called low phenylac, which is low in phenylalanine, that you can use while they're nursing. And then when they get older and are eating regular foods, they have to carefully monitor their protein intake to make sure that they're not getting too much phenylalanine. Foods that don't have much protein aren't really a problem. So things like fruits and stuff, you can eat as much as you want. But pretty much for your entire life, you have to be on this monitored low phenylalanine diet if you have PKU. So that's just one example of autosomal recessive disorder. The other one we're going to talk about is cystic fibrosis. And cystic fibrosis is also due to a mutation in a gene that makes a protein. With PKU, the protein was an enzyme that was defective. In cystic fibrosis, the protein that is defective is a membrane channel protein. Remember how we talked about there are proteins inserted in the phospholipid bilayer that control movement of things in and out of cells. And in this case, the defective membrane protein is a channel protein for chloride ions. Like if you eat salt, you have sodium and you have chloride. And so this channel controls the movement of chloride ions out of the cell so that and outside of the cell, they're important in controlling like osmotic pressure and the composition of fluids, of how much water. And this mutation is a particular problem in tissues that produce mucus. So here's like a layer of mucus on top of on outside of the cell. And what happens if the chloride ions are not present, then the mucus that's formed is very thick and sticky. So tissues or organs that produce mucus as a normal function, the mucus they're producing is very sticky and thick and can clog up organs. So the places that this is particularly a problem are the airways, like the lungs and the bronchial tubes, where you get this buildup of thick mucus, which can lead to breathing problems, lung damage, um, frequent infections, because the bacteria and stuff like to grow in that thick mucus. Sometimes people with, especially young children with cystic fibrosis, have to have the mucus sucked out of their lungs to remove some of it. The intestine, the digestive system, you also get clogging up of mucus, so that the enzymes involved in breaking down your food cannot get to your food properly, and that can lead to problems with digestion of food and absorption of nutrients. And then in the reproductive tract, particularly in males, there's mucus in the pathway that sperm take as they travel through the reproductive system. And that, again, that thick mucus can cause problems in sperm movement and can actually even lead to sterility in males. There's also accumulation, blockage of sweat glands. So those are the major problems associated with cystic fibrosis. There's not really a cure. There's just some treatments to help prevent more serious damage. Okay, so what we've talked about so far are disorders on autosomal chromosomes. So that's chromosomes 1 to 22. Everybody has a pair of those, one you got from your mother, one you got from your father, in all of your cells. Now we're going to switch to talking about the sex chromosomes. And the sex chromosomes is the last pair. It's the 23rd pair. And in this case, not everybody has a matching pair, because females have two X's, but males have an X and a Y. So the genes on these chromosomes, females have two copies of them, so they have a homologous pair. Males do not have a homologous pair because they only have one X and one Y. So they only have one copy of those genes. These are called, the genes that are on the sex chromosomes, particularly X, are also called sex-linked genes. And the X chromosome is much, much bigger than the Y chromosome. This is a picture of an X chromosome and a Y chromosome. So you can see how much bigger the X chromosome is. The Y chromosome, for the most part, only has genes related to the development of male characteristics. So whereas the X chromosome, it does have genes related to the development of female characteristics, but then it has a lot of genes that have nothing to do with sexual development. So for those genes, females have two and males only have one. And we're going to talk more about them. The sex chromosomes, your inheritance of sex chromosomes determines your sex. So if you have a Y chromosome, there is a gene on the Y chromosome called the SRY gene. And at about 6 weeks after fertilization, it is activated and it turns the kind of embryonic gonad into testes. So before 7 weeks, you're undifferentiated, your gonads. They're neither male or female. So this would be kind of the embryonic gonad. And if the SRY gene is activated, it produces a protein that converts the undifferentiated gonad into male testes. If that gene is not activated because you don't have a Y chromosome, but you have two X's, kind of by default, the embryonic gonad turns into ovaries. So it's the activation of that gene around 7 weeks after fertilization that controls the sex development of the embryo. The outcome of a particular mating can be even looked at by a Punnett square, because females only have two X chromosomes, so they can only give their offspring an X chromosome, whereas males have one X and one Y. So during meiosis, when they make sperm, about 50% will have X chromosomes and 50% of their sperm will have Y chromosomes. So when fertilization occurs, it can either be a sperm with an X chromosome or a sperm with a Y chromosome. If it's a sperm with the X chromosome, you'll have two X's and the offspring will be a female. If it's a chromosome with a sperm with a Y chromosome, the offspring will be a male. So you have about a 50-50 chance of having either a female, because these two would be females, or a male. And it's really the sperm that determines the sex of the offspring, because females only have X's. So it's whether or not that egg is fertilized by a sperm with an X chromosome or a sperm with a Y chromosome. Because the X chromosome has a number of genes that have nothing to do with sex determination, males only have one copy of those genes, whereas females have two copies. So males, because they have an X and a Y, only have one copy of genes. Females have two copies. So if a male gets a defective gene on an X chromosome, they essentially can't be a carrier because they only have one X chromosome. So with a defective gene on one X chromosome, they have that disorder. Whereas for a female to have that disorder, she has to inherit it from both parents, the defective gene, because she has two X chromosomes. So therefore, because males can either be normal, so they have a normal X and a normal Y, or they can be effective. And for genes that are on the X chromosome, they use this indication of an X with a superscript. So this would be the defective chromosome X, and then this would be the normal one. So this means that this male has a defective X, a normal Y. So that means he would have that disorder. And only if he had an unaffected X would he not have it. With females, they can have two normal. They can have one affected and one normal, which would make them a carrier. So they wouldn't have the disorder, but they would have a defective gene that they could pass off to the offspring. The only way they would have the disorder is if they had two affected chromosomes, which means they got one from their mother and one from their father. So a female can either be normal, which is this one, be a carrier, which is heterozygous, or be affected by having two defective genes. So this is just showing that if a mother is a carrier, and this would be normally the case, and the father is not affected, that there is 50% likelihood that both you would have normal children, 25% that you would have a carrier girl, and 25% that you would have an affected boy. And you can kind of gather from this that sex-linked disorders are more common in males. And they're more common in males because they only have one X. So that if they get a defective gene from their mother, they have the disorder. whereas a girl has to get it from both her mother and her father, which means her father has to have the disorder. So that all of the sex-linked disorders, or X-linked, that means the same thing, are more common in males than females. Oops. So I'm going to briefly talk about three sex-linked disorders in humans. And these are all due to defects on the X chromosome, of a gene on the X chromosome. So they're all more common in males than females. Color blindness, muscular dystrophy, and hemophilia are the three I'm going to talk about. And color blindness is more common in men. There are a couple different types of color blindness, but the one that is it's on the X chromosome is the ability to distinguish between red and green colors. And that is due to a defect of a gene on the X chromosome. And so individuals with red-green color blindness, that's usually diagnosed by using a series of plates. Some of you may have had a test done with this, where a doctor could show you various pictures and you would have to be able to distinguish, like in this one right here, there's an 18 embedded in the green, a 17. So if you wouldn't be able to differentiate these numbers or letters in a colorblind test, that would be indicative of red-green color blindness. This is not a particularly fatal, it's not a fatal disorder, but it does affect your capacity to distinguish colors. You can still drive because besides the red and the green light being colored, they also are always in a particular order. So you can tell that which one is, whether it's a red light or a green light, without being able to see colors. A much more serious disorder is hemophilia. There are a couple different types of hemophilia, but the more common type is on the sex-linked on the X chromosome. So again, it's much more common in males than females. Hemophilia is a disorder in blood clotting, so that if you have hemophilia, you're not able to form blood clots. This is a great picture. It's this one here. It's a scanning electron microscope picture of a blood clot, and the little red disc are red blood cells, and then the clot is formed by this protein fibers that kind of form a spider web that traps the blood cells, and that's what actually forms the clot that would close up an opening. So if you have hemophilia, if you get any type of injury, either external or internal, you're not able to form a blood clot, and therefore you have serious problem of uncontrolled bleeding. So here's a picture of, it doesn't have to be an open wound, like a bruise indicates bleeding under the skin. And so if you have hemophilia, you're not even able to stop bleeding under the skin. And here you can see the accumulation of blood swelling up that little child, probably little boy's elbow, because of not being able to clot the blood. What else was I going to say? Hemophilia is treatable. When they first started being able to treat it, because it's a serious disorder, even things like having dental work and stuff become much more serious, where you might have minor blood loss, but could be very serious if you can't form a clot, was treated with blood transfusions to get clotting factors from other people, so to replace the missing clotting factors. And there was actually a time period before they started testing blood supply for HIV that some hemophiliacs got HIV from their blood transfusions. Now they can isolate the clotting factors a little bit from blood and can administer those directly. And so it's a protein involved in the clotting mechanism that is defective in hemophilia. The third sex-linked disorder that I'll mention is muscular dystrophy. Again, that's more common in males and females. And this is due to a defect in a protein in muscle tissue. And so you get abnormal looking muscle tissue. This is a normal muscle cells. And you can see all this like fibrous buildup within the muscle. And so as a child with muscular dystrophy gets older, their muscles get more atrophied and they get increased weakness. These are actually not the same boy as they are growing up, but it's actually 5 brothers, all of whom have muscular dystrophy. And you can see that the littlest guy here looks pretty normal, but you can see with the bigger ones, their stance is abnormal. They're trying to compensate for their weak muscles to kind of change their posture to be able to hold themselves upright. So they get continued deterioration by muscles, and usually by the time they're in their late teens are wheelchair bound because of muscle atrophy. So that's the third disorder associated with sex-linked genes. And again, that one also is more common in males than females. Since this is getting pretty long, the PowerPoint, I'm going to stop here and make a third genetics PowerPoint where we'll finish up this topic.